So we are going to finish with this. I tried to break the class because they has a lot of molecules, and sometimes it could be a little bit overwhelming to talk about so many things at the same time. So now I'm going to talk about how the regulation of the G proteins. I'm going to keep only the pathway, one pathway for this class, and I will finish with this other type of regulation. Protein stuff are not, it's something that is very, that seems to not have any implication in your life, but actually, especially if you're going to get it in healthcare, it's going to be, you are going to be overwhelmed by this. For me, it was a surprise that actually the percent that I taught this, I didn't know, and the students didn't know this. Duncan, do you know, have you heard about OxyContin?
Yeah.
Could you tell me a little bit, what is that?
It's just like a painkiller that lots of people have got addictions to.
Yeah. Do you know what is Purdue Pharma?
Not specific.
Somebody knows what is Purdue Pharma? Yeah, that is fascinating. That because, have you heard about who is El Chapo? Yeah, everybody knows. Maddie, do you know who is El Chapo?
It sounds familiar.
It's a drug dealer that right now is in the States. So Purdue Pharma was like El Chapo, but legally. And now it's incredible. You cannot even Google the picture of the people because they bribe so many people in the States. You know who is Rudy Giuliani? Rudy Giuliani. One of the main supporters and the lawyer of Donald Trump. He was one of the lawyers of Purdue Pharma. So you see the level of corruption that they had. So I'm going to show you some videos. Painkillers could be addictive. Yes. So for example, one day, hopefully, some of you will be doctors. Some of you will be veterinarian. So for example, if I ask, you will see that, for example, if Julia become a OBS obstetrician, yeah, and she will have to give painkillers to moms when they're going to give birth. And I don't know if all of you, for example, Derek work in oncology. He will definitely have to give painkillers, opioids to patients that are going to die, yeah, to improve the life of some of the in the last stages, and they are wonderful for those people. However, I will show you that the point is that how much money do you make of a medication that is only target to use for few people? However, what about if we use that same medication for any type of pain? Yeah, you would make millions of dollars. So that was the problem that they have, and I'm going to show you a little bit about that. So very good. They used to call them not OxyContin, but OxyContin, because so many people died, and this is a name that they give it in the street. And
we're going to look at one of the major causes of this crisis, prescription painkillers, which are chemical cousins of heroin. Around 75% of heroin users started with a prescription opioid, drugs like OxyContin, Vicodin and Percocet, which some take recreationally and others take as prescribed by a doctor. But however it starts, it can get out of hand fast.
So how many, if we start from this side, have moved from prescription drugs to heroin? Everybody.
And heroin's so much more cheaper.
Once people became dependent on painkillers, they said they'd do almost anything to get them.
One time I was hurting so bad that I ended up punching a 4x4 and breaking all 3 of these fingers and this bone to get pain meds from my doctor. I did that several times.
So you'd hurt yourself in order to go to a doctor?
I had 9 teeth pulled out of my head for prescriptions.
At the time, that's how
to get a prescription.
Yeah. I've been with like groups of people where they've taken like bats to their wrists,
broke arms
easily.
I mean, how many people do know someone that's died of overdose from? Everybody.
This is happening everywhere. The odds are right now you probably know someone who is struggling or who has died from an opioid addiction. Some towns have been devastated. Just 2 months ago, in and around Huntington, West Virginia, 28 overdoses occurred in one 5-hour span. And given their risks, it's kind of amazing how freely doctors have prescribed them, because there are a lot in circulation.
We have currently nearly 250 million prescriptions for opioids written every year. That's enough for every adult in America to have a bottle of pills and then some.
Holy shit!
Okay, so we talk about that there is a ligand, that there is a receptor, that receptor that is coupled to the G protein, that the G protein has an alpha and a beta and a gamma components, yes. And we talk about second messengers. Now we're going to add more complexity to this. You have also effector proteins, and you have large effector proteins. And then you will have a cellular response. So what are the effector proteins? Effector proteins are molecules that act downstreams of the signaling pathway, so they are below the ligand, the receptor, and the G protein. Here they are downstream, and they will be activated by upstream ligands. So here in this case is norepinephrine, the one that is activating that. And these effector proteins, you will see that they are generally enzymes. So this adenylate cyclase is an enzyme that produce this cyclic AMP. Okay? And the cyclic AMP will be a second messenger. So as an example, I'm going to tell you about this. This is an example of an effector protein. And here you see that this enzyme here, so the G protein is activating an enzyme. And the next class I will show you that G protein generally activate other molecules, and generally the effectors generally are enzymes. And this one will be activated by the G protein, and that there is a alpha subunit that is going to activate this enzyme, and as a result of that, they are going to turn ATP into cAMP. Okay, so Asher, if I want to turn ATP into cyclic AMP, okay, so adenosyl monophosphate, what I have to do?
You'd have to get rid of 2 phosphates.
Pretty easy. So you have, we're going to go from ATP to AMP. So what the enzyme is going to do is, we're going to have different enzymes, and we are going to be targeting these 3 phosphates, and we're going to make also changes here. So here, we're going to get rid of these 2, and here we are going to have already AMP, and we're going to have one enzyme, the adenylate cyclase, that is the one that we were talking, and we keep only one phosphate group, and here, in this way, we have the cyclic AMP. If we have another enzyme, we turn it into adenosine monophosphate. So this one is the one that we use for energy, and this one is the one that we use for energy. This one is the one that is the second messenger. Good? So second messenger, the one for energy, and this is the one for energy. Clear, Maddie? These are the key molecules. Of course, in the final exam, if we were done biochemistry, you will have to memorize this one. these molecules. Here you just have to know the names. So here there is only one phosphate. Here there's one enzyme, here there's another enzyme, and here water get in, and we will talk about that. So cyclic AMP is the second messenger from the ATP using that enzyme that I told you, and that one is a very important signal transduction, intercellular signal transduction. If you one day Google CMP traffic or signaling, you will find hundreds of articles, okay? So, Angus, I didn't show you, unfortunately, the answer this time. I am waiting for the final exam, so you can have many answers there. Could you help me with this question?
What is cyclic AMP? It's B.
B is a second messenger, important for many biological signaling pathways, okay? So perfect, and we just talk about that. Derek. So here we have, how is this called? This pink stuff?
A receptor.
It's a receptor, so you see an R, and this, what type of receptor is this one?
Ligand binding receptor.
It's a receptor that is associated to this green stuff. Yeah, so it's a yes, receptor couple G protein. Yeah, all those things that it always is there. Page. So here we have the enzyme, this and this. Yeah, and the receptor. How we turn, I always show the domino. How I turn on all these things. What is the first thing that you have to turn on all the system?
I don't know.
So to everything start working. What is the first step? How we turn on the receptor?
Bind to it.
So we need a ligand. Good. Next step, Julia. What have to happen to turn on? So we have the ligand. What have to happen with the G protein to turn it on?
Would it also have to bind?
Sorry?
Would it also have to bind?
It has to turn the GDP into GTP, and it will dissociate, okay? I know that this will be repetitive, and then you will get it, okay? And Jenna, so ligand dissociation, how we turn on the enzyme here? So it has to, this alpha will separate and this beta and gamma will associate here, okay? So let me show you. So to turn it on, first thing, as Paige told us, you need a ligand there that it will turn on this. Then as Julia told us, we will need to dissociate the G protein into two, and we will need to, when this is dissociated, have to connect, has to attach to the enzyme. Okay, so here one key step is to turn this GDP into GTP. So first step, what Paige told us. Second step, you see what Julia told us. Instead of GDP, GTP, and you see these two work together. Sorry, I said that it was the beta and the gamma. The beta and the gamma are always together, and the alpha, it always go along, and it's going to, this is what Jenna told us, is going to attach here to the enzyme, and it's going to, this is what Asher told us, it goes from ATP, remember, has 3 phosphates, and it will turn into cyclic AMP. Good? So we turn everything on, and all the pathways will work in a similar way. Ligand, GTP, dissociation, and we turn on the enzymes. Okay, we turn it on here, and turn it on here. So now is everything working? Felicia, what happens if this is all the time on? If you have you're producing cyclic AMP all the time? That the organism is going to die, yeah? You have to, it's like we're going to talk about 2 diseases tomorrow. No, Wednesday, sorry, I'm also very tired. Cholera. What happened here is that they don't turn off the cAMP, the cyclic AMP, so people start losing a lot of water and they die because of losing of water and ions. And there are other diseases that you have that. So think about that. What happened if you put all your clothes from the laundry to the dryer, and instead of putting it 20 minutes, 40 minutes, you put it for, I don't know, 60 days, all time on. So they will be definitely, a fire department will come to your place, yeah? So same thing happened here. You have to turn on and off this mechanism. Good? So you have to turn it off. So could you tell me how we turn it off, Felicia? What steps do you see there that are happening? So what happened? What had to happen here? What had to happen here? And what had to happen here?
The receptor lets go of the ligand.
So the receptor lost the ligand. Yes. Very good. Yes, this is what you said.
Yeah.
Maddie, what happened here in the G protein?
The GTP turned into GDP and goes back.
Yeah, perfect. And the second thing that happened is that they came together back. So the alpha went back and is not there. So you will see that this is going to, if you understand this, you will understand most of the ways in which the G proteins work. Then you will change the enzyme, and you can change some names of the subunits, but it's going to be very similar. So here we turn it off, here we turn it off, and here we turn it off. Yeah, so ligand has to go, done. The G protein has to go to GDP, they reassociate, so this is empty, and this is how you turn it off. And this is very important, yeah, because they have to be short, they have to be on only for a short time, and they have to quickly change to adapt to different conditions. And we will talk about some diseases in the last class, but the idea is that this alpha G subunit need to be inactive to stop making cAMP. This is just an example of all the functions that can be involved in the cyclic AMP. It's hundreds. This is related to diseases, to the immune system. If you have all the time on the cyclic AMP, so type of cancers are also taking place. They also the cyclic AMP is also in form of the production of mucus in the epithelial cells. These are examples of that we have talked some of them in class. So forever, I think we will remember glycogen, and especially Angus will remember glycogen, and hopefully appear in his final exams of Cells and Energy, that the epinephrine is breaking that in the muscle and the liver. I also told you the cardiovascular, the heart rate, the epinephrine is also doing this kind of thing. So the epinephrine is going to go to the receptor, G protein, cyclic AMP, and this is controlling that. It also, here you have other type of hormones that affect kidney and bones, and those one are depend also of the cyclic AMP, and you will find so many more cases. Okay, so we have talked about up to here, up to the second messenger, and now we're going to talk about large effectors, and we are going to talk about the protein kinase A. They will be protein kinase A, C1 there will be hundreds. Here I'm just going to show you one example of what happens. So this one, these large effectors are proteins that function as a center of a hub. From here, you can have hundreds of different pathways that will have functions. The characteristic is that they're multifunctional. In other words, it's like an allosteric protein. It will have one thing to turn it on, and it will activate one pathway, or it can activate another protein. So this is why they are hubs, because they will be controlling different parts of the pathways. And they are generally related to signal amplification, as we said before. Here, we are going to talk about proteinase kinase in terms of the cyclic cAMP. That cyclic cAMP is going to turn on the protein kinase A, and is going to regulate a lot of different processes. Okay, so this is the protein kinase A, or PKA. So here again, so Maddie, 4 components. Could you tell me which one are the 4 components?
The regulatory subunit
Mm-hmm.
and the catalytic subunits.
Okay, so 4 subunits, 2 of them that are catalytic, 2 that are regulatory, and here the cAMP is going to go and attach. The cyclic cAMP is going to attach to a regulatory component. It helped me to remember that enzymes are like molecular scissors that are cutting different other molecules. And so if the regulatory components are like the hands, yeah, they tell exactly when they have to be on and off. Yeah, here the cAMP are going to attach here, and they are going to separate. And I will show you that in the other slides. And here, this is going to have a conformational change. So you will see that in the textbook, they will show that this will not look more like a square. They will have a change of shape, and that means that it will be active. Okay, so I just told you that this is a key protein that attached to a cAMP, the cyclic cAMP, and it attached it phosphorylates as usually this part of other proteins, and it's used ATP as all the kinases. And in your textbook, they tell you that there are 2 main components. There are catalytic and the regulatory components. And this one, the regulatory components inhibit this one. Right now, this enzyme is not working. You need to first, if there is no cAMP, is not going to work. So what is going to happen is that those one are going to attach there. And so here, when they are not attached, they look like this. When they are attached, they will start like this. They will start looking like this. Yeah, the cAMP is there. And now the next step is that it's like scissors that have a cover. Right now they have a cover. As soon as the cyclic cAMP is there, they are going to deattach, and there will be a conformational change. So the cyclic cAMP is there. So this is going to produce a change in conformation. And here you can see the change in conformation. Yeah, so now it's like the scissors don't have the cover anymore, and the enzyme is going to work. Well, and those 2 are separated. So I show you that the catalytic subunit deattach, and they are now activated, and they can phosphorylate the target of the cells and have multiple responses. Here is again the same thing that I have been talking from in the previous class, ligand, and here this is just a summary that you want to study, the G protein that attached to the enzyme, to the first effector, then the second messenger, and then it attached to the large effector that is, in this case, will be a kinase, and there will be a cellular response. And one of the responses, I told you that this one have many functions, that it one of the large effectors have multiple functions, and one of them, it will be to turn off this. Yeah, so here, these protein kinases are also cAMP dependent kinases, yeah. And here, one of the functions that it will have the cellular response, they will have many, but one of them is turning off this. It was on, now it has to be off. If it's on all the time, it can produce a lot of problems. So this is an example of a negative feedback. It was on, now one of the responses is going to be to turn off everything. Okay, so now the G protein is not going to work. So until we have a new ligand or we remove the inhibition. Okay, Maddie, we have made it up to here. We are still awake. Can you help me with this question?
ATP.
What activates? What activates the protein kinases? So the cAMP is going to activate the protein kinases. Yeah, I think it's well phrased. Yeah, so this one is going to activate that. I don't know if you said that. You said that. So remember, for the test, cAMP is going to activate this.